Okay, so we're back. Thank you for still being here for session 2, historical and emerging methodologies. So this afternoon we will be with Erika, Cassandre, Isis, and Victoire, and your jury members will be so Marie-Yasmine Deschraoui-Botin online from the lab Ecosys, Paola from IRCAN, and Daniel from IRCAN, and I'm here as chair only, but not as a jury member. Okay. Ready to go?
Yeah.
Allez, enjoy. If you can.
So, hello everyone. I'm Erika Alexandra Quiñones Bonilla, and today we're going to explore the past, from the past to the present of the ocean, with a comparative analysis of the ancient and the current global distribution of sparse aurata, merluccius, and different precious larvae. So, the ocean is a key source of food. Ecosystem service has sustained populations through the story. But this ecosystem service are threatened, and this is due to the climate change, which is altering the whole ecosystems, the biodiversity, and the growth and survival and productivity of some marine organisms. These changes are leading. Oh, I'm just going. Sorry.
You go ahead.
I'm just going to.
Yeah.
Okay, sorry. So the climate change is leading some distributional shifts, and with the results of changing some potential fish catches. So we know that fisheries and agriculture are facing many challenges, and that's where the ecology takes place, because the scientists are trying to investigate the future distributional patterns, how the dynamics of the ocean are going on, and etc. But the marine ecosystem, that are complex, of course, it's not presenting changes in the present. The dynamics of the ocean have been shaped natural and human factors over the story of the humanity and the earth. And that's where the anthropology takes place. So during my immersion project, the idea was to merge these two sides. Going to the past help us to understand how the present is it. The exploration of these anthropological subjects and data might help us to gain a better understanding of what's happening here in the present. So with that, we developed a question of research. Our question was, does the global distribution of the main fishes vary since ancient times, and if it's accelerated by the climate change? So to solve this question, I established two hypotheses. The first one is that the global distribution patterns of the main fish species changed significantly from ancient times to the present day, and also that these changes are related with the increase of the latitude in the distribution patterns of the fishes of the European world. So having the hypothesis, I established also two main objectives. The first objective of my immersion project was to showcase the global distribution of the main fishes species from the Paleolithic to the Roman period. And the second objective was to determine if there are differences between the ancient and the current distribution patterns. So how I did it? I developed and I conducted an extensive literature review during 4 months. This literature review was focusing on extract data of archaeological records and compilate the ancient catch data from the antiquity. It means from the Paleolithic period to the Roman period of the Atlantic coast of Europe. After having the extraction of the data of the Atlantic coast, we needed to gather with the available data of the Mediterranean Sea. In this case, I gathered this information with the data provided by CEPAM laboratory And also I included the current distribution patterns of the annual catches, and this data was provided by Ecosys Laboratory. So having all the data, the archaeological data and the present data, what I did was to transform it in from the archaeological data to the ecology biodiversity data. So first, I construct the global distribution from Paleolithic to the Roman period. It means that I have many maps. And also, I perform an analysis to the distributional centroid. And this analysis will help me to analyze the variation of the global distribution to the space and to the time. So let's see the results. I want to let you know that my results are in two groups. The first group is the literature review. I will tell you what I extracted from the data and how was the results and how was the process to extract data from archaeological records. And the other are the local analysis, of course. So during the literature review, as I told you before, I just extract data from the Atlantic Ocean, and 121 archaeological sites were identified. In these 121 archaeological sites, we could identify 6 different ancient periods and also 180 species identified. For our project, we just focus on the species that has the higher frequency. After, as I told you on the methodology, we gather all the information and the data available. So from the antiquity, we studied around 278 archaeological sites. And after merge all the data, we only select 3 species: Sparus aurata, Dicentrentius labrax, and Merluccius merluccius. But for this presentation, and we don't have enough time, I'm just going to be focused on Sparus aurata. So let's see the global distribution of Sparus aurata. Here, it was the distribution that we extract from the Paleolithic period. As you can see, only 2 sites were identified. Going to the Mesolithic, we could see that there are some homogeneous patterns into the Mediterranean Sea. If we go to the Neolithic, we can see that there were some presence of the Sparus aurata in the North Atlantic coast of France. During the Bronze Age, the trend was the same, concentrated in the east coast of the Mediterranean, and also we found a site in the Atlantic coast of Portugal. During the Iron Age, we can see the same pattern. The species are distributed around the whole Mediterranean, and also with some points in France, Spain, and also we can see here that some points appear in the Atlantic coast of the north of Africa. The Roman period, you can see here that the points are more concentrated to the west side of the Mediterranean, and then on the Atlantic coast, the points are on the France and the North Atlantic coast of Spain. This is the current distribution of the species, and this was provided by Ecosys Laboratory. And we can see that now the species is all abroad Europe, including the north of the Atlantic coast and the North Sea. So at the end, I was able to construct the geographical distribution of Sparus aurata from the antiquity to the present era. So we can see that the distribution is homogeneous through the Mediterranean Sea. It's concentrated the distribution of this species, but also there is an interesting distribution in the Atlantic coast. Currently, it's spread around the whole European and North African waters. We know that the variation of the distribution patterns is part of the ecology and the dynamic of the species. And here, with this study of the global distribution, we can see how the dynamics are presented through the period of times. Also, these dynamics are not only related with the time, also it's related with environmental factors that can affect the global distribution, and also the fish assemblages are influenced by human consumption. But in this case, and for this study, I didn't take into account the human consumption. We have the catch, but not the cultural subject, about the influence about the assemblage. So after having the global distribution of the Spinosaurata, I developed a distributional centered evolution analysis. This analysis can help us to analyze how was the evolution of the species through the space and the time. And you can see here in this graph. The x-axis of the graph, it shows the longitude, and the y-axis shows you the latitude, and the small dots with colors are representing the periods. So we can see here that I only take into account the Iron Age, the Roman period, and the current data. And this is because, as you see before for the Paleolithic, for example, I just have it 2 points. So we decide just to take into account representative data. So here in this graph, we can analyze that all distributional pattern of the species indicates a possible migration to the north, and also a decrease on the longitude. Here we are in the Mediterranean, so we can see that the Spinosaurata from the Iron Age that came from 800 years before Common Era, it's just going to the west coast, and also it's going up to the north. After having the results, what I did was to go to the literature and see if my results were having correlated with the results of other people. So yes, I found that the expected distributional center shift gradually increased over time. So it means that from the antiquity, sorry, and it's it was me. So the results and the literature mention that the distributional patterns of the species, it seems to be increasing over the time, not necessarily under the concept of climate change. Yeah. And concerning the Mediterranean Sea, that in this case we are here, we can see this author say that it's projected a northwestward distribution shift for cephalops, which we made the cephalops, and also demersal fish. So we know that this study that came from archaeological data recorded in archaeological sites excavation are correlated with the literature review. Also, Emma Bellamy, which is a person that works in ecosystems, she said that from the Mediterranean Sea to the future range shift from the Mediterranean Sea to the North European coast in the distribution of all the species for all levels of warming. So in this case, I didn't take into account the environmental conditions, just time and space, but we can say that the trend in our results are related with the results of other people that are making another studies and including environmental conditions and etc. So to conclude, what about the hypothesis? Well, we could see that there was a shift on the global distribution of the species study. And also we could confirm that an increase on the latitude range have been evidenced. As we know, we saw that possible migration of the species to a higher latitude was evidenced, and also that Spinosaurata from the Iron Age to the Roman period has a decrease in the longitude and an increase on the latitude distribution. So what about our question, our research question? Did work global distribution of main species percent variation? Yes, we saw it in our data and we correlated with the data from the other scientists. And it is accelerated by the climate change. I'm sorry. Probably. Yeah, I'm sorry, it doesn't appear. So we can conclude that going to the past help us to understand the present and also the future. From archaeology to ecology, we can say that using two different files in science, we can merge and understand how it's the dynamics of the ocean. Even if our data was not related directly with biodiversity, we just used ancient catch data. It gives us a good impression, and we are close to the results of biodiversity and on the present. So transform of archaeological data in ecological data is a preliminary result to understand how the dynamics are and how can we apply to management the future under the global environmental conditions. During this study, of course, we had limitations. The lack of data is one of them. Spending time exploring old books, trying to extract this kind of data, it's not easy. The reports are not in the same language. Also, the studies concluded by the archaeologists are not the same. The methodologies are not the same. So it's difficult and it's a limitation to understand the big picture of the archaeological data. Future studies are going to conduct. I remember that this Immersion project, it was made under the framework of Merrimack project. So to go further, it's necessary to include environmental parameters, also determine the numerous or species identified, the size of the fish that were collected, and also include distributional models and general circulation models. So I just want to thank you to Ecosys Lab, especially to Virginia Raybot and Emma Benlamy, also as a PAM laboratory, Tatiana and Gabriel, which helped me a lot to understand this archaeological data, and of course to the Mars team. I remember that this project is funded by the European Research Council. And just to going back to my conclusion, yeah, from the antiquity to the present, changes on the distribution patterns of the main fish species in European waters have changed. And the problem are not always the changes. The problem is how fast they are happening, because the species don't have time or enough time to develop adaptation strategies. Thanks.
Thank you, Erika. Do we start with you, Aldine? You just arrived.
Okay, so thank you so much, Erika. Beautiful job. Can you go back on your data collection slide? So I have a few questions regarding on that. So first of all, when you are looking for this data, how are you dealing with language?
Yeah.
And changing the name regarding the culture?
Yeah. So I worked in a big database that is now available. The name is the MArchive, and in this there's a first draft when you put exactly the data that you found on the papers or on the web or etc. So you find a record, and normally the record say the species and the number of species identified, and the number, sometimes it has the size and the type of methodology that they use. Yeah. So you put it there, and then after we created like a big data set with the species that we find, like the number that we found it, and the data set will give you the scientific name that it's currently accepted. So what I did is, for example, I found, I don't know, an Sparus, yeah. So I go there, and if it's not in that database, I have to search in the web to see what is the name that it's accepted now.
Okay.
And then I will fit the.
Okay. And then you are using also the common name and for the different country and.
Yeah.
Okay.
Yeah.
Okay. So then in England, I mean, you have a lot of data from the current distribution of Sparus aurata.
Yeah.
How can you comment on that? So, and you don't have, I mean, so many data from archaeological study, but so many archaeological studies have been done there in all the coast. So why you never integrate them?
Yeah.
And can you comment on that?
Yeah, I was asking the same because I was finding a lot of archaeological sites in England. I was like, what's happening? Because I'm not finding a species there. So there could be three main reasons. One, that I passed through that, it was that my study was from the Paleolithic to the Roman period. So the archaeological sites that are there in England, normally it's medieval.
Okay.
So it's after the Roman period.
Okay.
That's one. The other is that some of the areas of the England were submerged like long time ago. So it's possible that this data is not available because it's now submerged. And three, one is the type of analysis that the archaeologists make, because some fish ponds are really small. So there are a big possibility that in that kind of excavation, a lot of bones of fish were lost. So yeah, that was.
Okay. Okay, thank you. And maybe last one. So when you perform your graph, how many? You have only one, I think.
The central.
The distribution. So how many data is required to perform such study and have a really representative graph statistically?
Okay, when I asked it to the ecologists, they told me that to having the distribution, like the global distribution of an species, it just required 1 point, like the limit points that are in the border. So if you found a point here, it means that the species was here. So you can say like the global distribution is going until here, because it's the point, the limit points. But in the case of the shift distribution, what we did was to at least count 14 points.
Okay, to have something.
Yeah. And that's why I didn't put here Paleolithic and Mesolithic, because it was less than 14 points. Yeah.
Okay, thanks.
We have to move to you, Paola. Then we will go to Marie-Yasmine and Robin online.
Thank you very much, Erika. It was a very nice hypothesis-driven presentation. So congratulate for that. I have two main questions. The first one is how did you analyze your data in the framework of the evolution of the Mediterranean Sea? Because on this time lag, as you said in the first answer, that you have modification also on the sea level. You can also have changes in the geographical area of the Med Sea, also on the Atlantic coast. So how you can implement this information in your model and in your interpretation?
Yeah, okay. I'm not going to, I didn't create a model because I wasn't able to have environmental data and all the data to implement these kind of things. So in my study, it wasn't taking into account that kind of things, like how it was the level of the ocean and the shape, or if there were submerged or no, of how was the cultures in these places where I found the data. That's the people that are continue working on this project is going to deliver, because my data was one of the first steps. But I can say that the Mediterranean, for me, in my case, if I'm right, it's more stable than the than the North Sea. You know, there are some small islands that could be affected more than the Mediterranean. So yeah, in this case, I don't know if I'm answering you.
Okay.
Kind of.
But this moving from the east of
Yeah.
west,
For me, what I was thinking, like just thinking and analyzing, is that the species are migrating outside the Mediterranean. And that's one of the conclusions of not going outside, but they're migrating to the north. And that's one of the conclusions that Emma has in one recent paper that she published. But I think that that's why I can see that the longitude is decreasing. So the fishes are going here, go up. It's just
and do you have an idea of some other methodology that can allow you to see this flux of population, this movement of population?
yeah, of course.
You use the archaeology, so it's a very good way to answer to some
Yeah.
question. But do you have some other? Do you know if there is other tools to go
Yes,
back to the past?
To go back to the past, for the moment, I think that there is only the archaeological records. If you're going really going to the past, and now it's just not the catch, but also the historic data of environmental factors and abiotic, but they're not that far away, so it's difficult to do it. But there are also some. I found some data in records, not of archaeological like records, but also where the people are telling stories about recipes or telling a story about how was the interchange of species. So you can find information there. For example, I know that some of the data that I have here, it was in the amphoras of the Roman period, so it was inside the sauce. So yeah, that's just the only thing that you can find, because there are not records. Also, there is the geologic analysis, when you can see like stratification of the sediments, and then you can find some another kind of things. But honestly, the studies that they are made in the archaeology, it's based on the archaeological sites. The people now are trying to redo it, like the studies, because the bones are safe, so they can just take again the bones and try to analyze and do it another kind of samples with ADN and trying to with the autolytics and other things to know if there are different species that before they weren't identified. But yeah, it's really difficult. Why?
Yeah, and in your mermaid project, there is some genetician guys.
Yeah, they're doing a lot of different type of analysis to determine the global distribution.
Because even with the current species, you can go back to their genetic history.
Mm-hmm.
So with current species, you can see some mark in their genome of where they come from.
Okay.
So you can reconstruct your population through the DNA of the species that are present today.
Yeah.
Full of passion, your answers. Thank you. So we can go online. Yasmin, I propose you to start the questions at your microphone is on, and then we'll finish with Romain. Maybe one, maximum 2 questions per person for this time. Sorry.
Okay, yes. Thank you, Rika, for this beautiful presentation, and also quite interesting. I mean, I really enjoyed this early interdisciplinary approach with archaeology. It's quite fascinating. I have really methodological question more, let's say, on this aspect. You mentioned a review. I would like to know how the data were. I mean, have you how the research of this literature was conducted, and were you part of it, or yes? Can you explain? I was lacking some information at this level and what you had in hand to start this project.
Okay, at the beginning of my immersion project, I went to the PAM laboratory. So I worked with Tatiana and Gabriel, they are archaeologists, and they explained me how the records are made and to understand it. And then they gave it to me a lot of books, and then I start with the structure. I merged and I compiled the data in a big data set, and also using Zotero to put all the bibliography.
So they
And
gave you the different books, and the references were already identified. It's in the literature
yeah,
review.
because they made already before that I started, they have it the Mediterranean data ready.
Okay.
So they gave it to me the Atlantic data, and also I didn't found like many. When I finished with the books, we started to contact some archaeologists from another archaeologist, and then they start to send us papers, and also of course in the web, Google Scholar, Science Direct, and etc.
Okay. Yes, and the other question I had was also maybe on the methodology, how the species, because you mentioned 3 species, then you selected one. What criteria did you use? Why prioritizing this one species?
Okay, I selected Sparsobrata because it's the species that has more reliable data to analyze, and also the other species. You know, when we made the frequency of the species in the sites, so we have it the species that we could analyze, and that species could probably be included in a model to develop a model.
Okay, so there are criteria, and okay, this was not, maybe I missed it in the presentation, or maybe it was not there, I don't know.
It was according the frequency and also
Okay.
the availability of the data to develop future models.
Okay, well, thank you. I'm sure it was a lot of work anyway.
Thank you, Maria Jasmine.
Yeah.
Thank you, Yasmin. Robin?
Thank you, Erika, for the presentation. It was very interesting, and it's always nice to go back in the historical data. I really enjoy it. I'm going to go very quick. I had a bit the same question as
Yasmin.
no.
Paula,
Lily.
Aldine.
You have no record of Sparusorata in Italy, where you do have some sampling sites. And I mean, there are very few, but there are less records than you have sampling sites. While I'm pretty certain that the species present there. Do you have any idea why? Is it because some sites are not? I mean, do you have any idea why?
Let me just see the distribution. Yeah. Honestly, my data is based on the archaeologic research. So yeah, that's a probability that there is not archaeological sites there, and that's why I didn't found data.
But there were some in the previous line. If you show like all the sites that you have, you do have sites in there.
Okay. Where are the sites?
I don't know. It was somewhere.
Do you remember the number of the slide?
What? I didn't get it.
Do you remember the number of the slide?
Yeah.
For me, it's the, I don't know, it's a result, the one with the 278 archaeological site. It's just before.
Okay, I understand.
This
Okay.
one. Yeah.
This one.
Yeah.
Okay, well, honestly, I don't know why there is no presence of a lot of presence of this data. But what we can say is that the presence in Italy it was established. So yeah, our results it's based in absence and presence. So I can say that in that country there were this kind of fishes. So the distribution patterns that you can see just state that this species is in the Mediterranean and in the whole area of Italy. Honestly, I don't know if the Adriatic Sea, maybe it wasn't different with the conditions before in antiquity, and that's maybe a point to take into account. But yeah.
Okay. And can I ask a very small...
Yeah, last question, go ahead.
So you look at, I mean, it's there are two questions in it actually. Did you account for the distribution of the sampling sites when you did the centroid analysis? Because given that you have a much higher concentration, or you seem to have a higher concentration of sites, basically in Sardinia and south of France and in some areas, did you average something, or did you just compute the centroid for all the different sites together?
No. It was this slide when you can see the distribution is of all the species. So it's not the distribution that you're seeing just of Asparus aurata. It's the sites, like the whole sites, okay? And when I did the centroid, I made a mean. It's like a mean of the distribution of the species per period.
But if you have, what I'm saying is that if you have many more sites in one location, it's going to give much more weight to this area, while it's actually the same abundance, slightly the same abundance because it's the same area.
Yeah, that's true. I just took into account the sites that the periods that were at least 14 sites. You know what I mean? 14 sites, and then I made the mean, and then I compute the longitude variations.
Okay. Well, thank you.
Thank you, Robert. We have a few, look at your screen, a few hello from former students. For the people, I just let you have a look. Sorry for the users, it's just personal, maybe. Floran, and also something, understanding the evolution in time and space of species is key to developing protection and climate change strategies. Well, I'm sure you know. Thank you, Erika.
Thank you.
Allez, Cassandre, here we go. Okay, so...
Yes.
Wow. Si je fais ça, c'est bon?
Okay. Is this
All good?
right?
Yes, this one. You can check if you want. Okay.
No spoiler.
There you go.
Okay. So when I was a child, I always dreamed about being an explorer and becoming, the person that discovers new territories. So I was super sad when my dad, who is online, told me that it's not possible anymore because of satellites. But well, it's not entirely true. So let me take you on this journey where I had to assess for the very first time, it has never been done before, the fish and coral communities around the Tatlong Polo archipelago in Oriental Mindoro, Philippines. To give you a little bit of context first, so my table of content, my presentation follows the regular structure of a normal presentation, but spoiler alert, my discussion will be about an MPA proposal. And my conclusion will be about what's coming next in the future. So, first of all, the Philippines are located in the Coral Triangle. So this is here on the map in Southeast Asia. This is the area of the world where there is the highest coral coverage.
and fish biodiversity in the world. So it goes from the Solomon Islands to Malaysia, and at the apex, the Philippines. So the Philippines is an archipelagic country, right? Meaning that it has more than 7,400 islands. It's the third longest coastline in the world. It shelters more than 1,700 reef fish species and 9% of the world coral reef. And so since it's an archipelagic country, people highly rely on reef for their local culture and economy. But as you know, reefs are currently threatened by global change and anthropogenic pressures. So this is why we have marine protected areas, so MPAs. MPAs are used to reduce local threats all around the world. And it has been proven that well-enforced MPAs and no-take zones, so MPAs where all kind of extraction is banned, are efficient to increase the fish biomass inside the boundaries and outside, thanks to the spillover effect. And this is where the sponsor of this Immersion project, Blue Finance, comes in. So Blue Finance and Blue Alliance are two sister organizations managing the MPA network in Oriental Mindoro in the Philippines. But where is Oriental Mindoro in the Philippines? So this is Mindoro Island, and this is the Verde Island usage. So the MPA network that Blue Finance and Blue Alliance manages belongs to the UNESCO biosphere. And the Verde Island usage right here is an area with a high importance, ecological importance for shorefish, but it's also highly threatened by anthropogenic pressures. So this is why MPAs are super useful in this area. And if we zoom in, there are 3 islands, a little archipelago right there in Oriental Mindoro. This is the Tatlongpulo archipelago, made of these 3 islands. And we almost don't have any data about these islands. So this is where I come in with this project. So the aim of the project was first to assess the fish and coral biodiversity around the archipelago, thanks to the fish visual assessment metric attributes, so photoquadrats analysis and reef complexity. We will talk about that later. And at the end, the goal was to use this data to design an MPA proposal. So the MPA proposal should mention as well the future ecotourism activity that they want to develop in the area, because there's currently no tourism at all. So the research question was, what part of the archipelago should be proposed as an MPA and why? Regarding the methodologies, so as I said, we almost don't have any data. So it brings me back to Les Reims. Where do I locate my stations for the monitoring, and how do I do it, and how many stations are useful? For that, since we don't have any data, Denmark Camera, the head scientists, came up with a methodology, the Mentato. So Mentato is a cost-effective tool for rapid visual fish assessment, and it's also used for chronophone monitoring, for example. So it covers long distance in a short time. So here is an illustration of me being towed behind a boat, because the principle of Mentato is to have a snorkel diver being towed behind the boat at a slow speed, of course, for intervals of 2 minutes. And then during the 2 minutes, the observer observes the main characteristics that we are interested in. And then after the 2 minutes, the boat stops, we record everything, we record the GPS location, etc. So here we wanted to know where are the biggest reefs. So we were looking at the hard coral coverage and the soft coral coverage. Then once we were able to look at the transect and the stations, which I will show you later, we move on to the actual data collection. So for the bentos, for the reef and the corals, we use the benthic photoquadrats based on Atrigenio paper that Blue Alliance used in the past, so we can compare the data at the end. We use 2 transects of 50 meters per station in scuba diving, so it's in shallow waters because it's shallow in the area, and because Blue Alliance use shallow water transect for all its monitoring, so we want to be able to compare the data once again. And so we take 1 picture with this device of 1 meter square of bentos along both transects. So 50 meters transect, 50 pictures. And then we we put all the images on CoralNet and we generate 10 random points. And this is the benthic attributes that we are actually identifying on the CoralNet. Then on the same transect, we have the fish visual senses survey. Same transect, same paper for the methodology, same shallow waters. We use this methodology to assess the fish biomass, richness, and biodiversity. We identified the fish at the species level, and I chose to focus on 193 species because of reason I can explain in the discussion later if you want. And we have to estimate the size super precisely as well to be able to have an accurate biomass, so at more or less 1 centimeter. And finally, maybe you never heard about that before, it's the reef structural complexity. We want to look at that because the reef complexity is actually the structure and heterogeneity of the reefs. So it has been shown in the literature that correlations exist between this complexity and the fish assemblage, so biomass, richness, etc. So we based our protocol on this paper right there that classified the structure of the reefs in 5 different categories. 0 here being the flattest reef ever, and the 5 here being with the most cavities inside. Regarding the results, we therefore still have to locate our stations. I still haven't shown anything about that, and then process the data collection. So this is the result on QGIS of the main taxonomy. This is the hard and subcoral coverage mapping along the transect. So in the lightest color, you have the lowest coral coverage between 0 and 10%. And in dark red, you have the highest coral coverage. So from 76 to 100% of coral coverage. And Blue Alliance actually conducted a few surveys before in those areas. So in medium coral coverage before, right? So because of that, because of the cost limitations, of course, because of the time limitation, and because of the physical parameter of the area, we chose to locate 11 stations for the following results and for the data collection. These are the final stations we chose to implement in the area. For the fish response census, I was able to identify 103 species out of the 193 that I knew for sure. So it makes almost 17,000 identification at the species level of individuals. So now let's have a look at the biomass density and species richness. Regarding the biomass, so this graph that you see here have been mapped on a map for an easy representation and spatial representation of the data. So you can see that the highest fish biomass was observed in Pulong Maliit Island, in the east of Pulong Gitnam, and in the south of Pulong Maliki. Similarly, we have the highest fish density, that is the number of individuals per volume, observed once again in the north of Pulong Maliit and in the south of Pulong Malaki. And finally, we have the species richness that is once again observed at the highest in Pulong Maliit, at the southwest of Pulong Malaki, but also in the eastern and northern side of the island. Now the photo quadrat results. So this is about corals, okay? In red, dark red, this is the hot coral coverage, orange soft coral, yellow fire coral. We want to look at the dead corals as well, so in white, and the abiotic attribute in gray. So over the 11 stations, we took 1,050 pictures, which makes 10,500 single points identifications, okay, of benthic attributes. So the highest coral coverage was once again observed here in Pulong Maliit. And then overall, it was with 40% coral coverage around the area, which I want to know here is that I don't consider the algae and stuff, okay, on the graphs. And most coral subcorals were observed in the south of Pulong Malaki once again, and the highest number of dead corals were observed around Pulong Malaki. Looking at the coral genus richness, The highest coordinate richness was observed in the south of Pulong Malaki and the north of Pulong Maliit again. And regarding the reef complexity, same. Highest reef complexity, so of almost 4 were observed in the east of Pulong Kitna, in Richards Point here, and on the north of Pulong Malakit. So this brings us to the MPA proposal. What do we do with this data? So the MPA proposal was based on this data collection, but also on the few assessments that Blue Alliance conducted before, and on other factors that we're going to discuss right now. So this is a picture of the first meeting. I presented my results during the city council meeting of Calapan City. Calapan City is the capital city of the island. Okay. So as we saw right before, the fish and coral assessment strongly suggested that the northern island, if you remember, Pulong Maliit and Richards Point and the southern island should be in the MPA if we implement the MPA. But the fisheries and tourism specialists did not agree with that. Why? Because they think that for the enforcement, it's going to be too complicated to go that far away from the coast in the smallest island where everything was at the maximum. And also the designation as fully protected area, because we want a fully protected area because of its effectiveness, right, in protecting the reefs. But if we want later to develop ecotourism in the area, we cannot design the area as fully protected because then no human activity would be allowed. And so it's going to be a problem for tourism. So we had to review the initial proposal, which gives us this final map. So here in red, you have the actual MPA that is proposed around the area. So if you remember, this is Pulong Malaki, Pulong Kitna, and Pulong Maliit, okay? The highest fish and coral diversity was observed here. Because as we said right before, for the enforcement, they believe it's going to be too complicated. Because if we don't have a well-enforced MPA, then it's an MPA that doesn't reach its goal of conservation, which is called the paper MPA, and we don't want that. So we chose to only these 2 areas of the islands. And why such small areas is because Blue Finance and Blue Alliance want to create an MPA network of small-scale MPAs. Because what's important to understand here is that we said that the Philippines is an archipelagic country, and therefore people highly rely on coral reefs to live and on fisheries. So we cannot just say that the whole area is going to be protected and that fishermen cannot fish anymore. So we have to consider that. And so I'm really glad and proud to be able to say that on the last week of the internship, this proposal that you see right here has been approved by the City Council of the Philippines. So yeah, new MPA coming in the Philippines. And so what's next now? The MPA will not be implemented yet because, as you know, probably there's a lot of paperwork and stuff. So still a long process before it's effectively implemented. But from this study, what we can learn is that there are healthy reefs, but future additional studies are needed, including for deeper reefs also, because fisheries are targeting deeper fish, like around the 30 meters area. So we should go study that area. And what I was really glad to see also is the eagerness of people and the local government to protect this area and their future. So it gives me hope in the future of marine conservation in the area. And now there are some other solutions compared to MPA possible. So for example, here you have a picture taken from the Blue Alliance LinkedIn of people working in the mangrove crab industry. So they try to launch a new project like that to reduce the fishing pressure. So even if we cannot have the whole area protected, we have less pressure on the fish stocks. And also it supports the local community. And also we still have to discuss the dive sites and the dive fees, of course, to generate sustainable revenues for the MPA network. Thank you very much, everyone. Salamat sa lahat, as we say in the Filipino, in Tagalog. And I'll leave you here with the MPA proposal.
Thank you, Kassan. Allez, let's start with the jury who is online, who had only one or a couple of questions last time. Maybe we finish by Robin, so we can start with Robin and then go with Yasmin.
Thank you very much, Kassan, for the presentation. It kind of looks like a dream internship, to be honest. I don't think it's everything's so successful it's long internship, but it's not that long to achieve so much. So it's really impressive, and it was very well presented.
Thank you.
I don't have many questions, I think, but so you said you identified 103 fish species.
Yes.
That was during the how you call it, the census, underwater census, or
fish visual census surveys. Yes.
so no picture, just while scuba diving. You identified that was while scuba diving, or it was taking pictures and then identify.
No, while scuba diving. So basically, we have a slate, underwater slate, and a pen, and you just take notes underwater of the fish you see, and then you enter all the data after the dive on Excel. So that's why I have this limitation of species that I could identify, because I have to know everything by heart. And for each species, there is a juvenile phase, the intermediate phase, and the adult phase that are completely different. So yeah, that was the biggest limitation for this phase.
But most of the species that are there are like relatively common species.
Yes. Yeah.
Okay. Just one remark. You were a bit fast on presenting the results, at least because you put these bar plots and you identified the zone with the highest densities for the biomass, the richness, etc. But there was no time to understand what the bar plot were showing, and the order of the x-axis was different from the order it appeared on the map. So it was quite difficult to understand that the left bar were for the island on the right, and then the one in the bottom was the bar in the middle. There was, because you were going quite fast, it was a bit difficult to follow, but at least, you highlighted the main message. So it's just a remark. You found some areas with low coral coverage, among other in the Pulong Malit island.
Yeah.
What was that due to? Because there seems to be some in almost all islands except the Pulong Malaki.
Okay.
That?
Yeah. So basically, I want to recall that this graph that you see here, like the plots, it's only for these benthic categories. So for the hot corals and all, and so it doesn't consider all the algae and stuff.
I was not thinking of this one actually.
Oh, okay. Which
I was
one?
thinking of the first one that you showed with from the Mantato.
Okay. Okay. So for Mantato, it's so this kind of map, right?
Yeah.
Okay. So Mantato, it's a methodology that is completely subjective, and the biggest limitation, I would say, is the water visibility, because the observer is on the surface. So here it's okay because we are interested in shallow areas for the reefs, for the assessment after. So it works, and the visibility in the area is around 6 to 10 meters usually. But on the day we did the Mantato, it was quite turbid. So in some areas, we believe that we made some mistakes of identification, and so we might have overestimated or underestimated the coral coverage in some areas because we couldn't see well enough. We were two observers for that. And so this is highly subjective. It gives a good overview of what the reefs look like, but this is not 100% true. Do I answer the question?
Yes. Well, because it seems like, partially yes. It seems like at least for the Gitna and the Malit, it's like areas that are mostly to the north. Is there any overall exposure to the ocean or to anything, or the opposite?
Yeah. So wait, I have in the annex the detail of the maps, because I have. So here it considers both soft and hot coral coverage, right?
Yeah.
And so it's here. Okay. So here you have a more detailed overview. So as you can see, it's the areas with the hot coral coverage that are the highest, and it's in Pulong Maliki. And indeed, there is not a lot in Pulong Malit. But if we add the soft corals on the right map, it adds more core coverage, obviously. And so that's true that the conditions at sea are like the currents are coming from the northeast, so from the small islands. It's more exposed, but it's also a bit deeper. So that's why I was saying that it was super subjective, because there are some parts where we couldn't see properly. So we just assumed it was rocks, while in fact it might have been hot corals, for example.
Okay, because it kind of not contradict, but it seems a bit contradictory with the fact that you have the highest fish abundance, fish biomass, fish everything.
Yeah, that's true. That's why I want to highlight that it's subjective once again. But there's no scientific explanation for this one. For the next, yes, but.
Okay, I may leave the floor for that.
Yaslin?
Yes, thank you, yes, for this beautiful presentation. And indeed, it looks like a dream internship with all the work you have been doing. And I again have, because it's not my field really of expertise, but again, methodological question, because it seems to be a lot of sampling, a lot of expertise. What's your confidence in the data you obtain? I understand that you mentioned that it was a subjective, but in terms of fish identification, I quickly read 1,000 photo, 10,000 item on CoralNet. I'm not sure exactly what this means, but can you tell me more? Yes, how much work it took and what concretely you have been doing?
Yes.
And how you analyzed, and what's the confidence on your results?
Okay. So I was, for this project, completely autonomous. So I had the supervision on site of Denmark Camara, which is the head scientist of Blue Alliance, but he's a fish expert, not corals. So for the corals, for example, I already had experience in coral identification in Indonesia from my past year, from the year before, during my bachelor internship. And so I compared my data that I obtained for the coral analysis. I based it on the papers and on the super great identification book that I don't have today, I'm sorry. And I compared it to the result that they obtained in other MPA in the area to see if it was relevant, if I would see, for example, one coral genus in a high quantity, in high abundance, I would maybe be surprised and I would maybe doubt my identification. But everything was correlated to the previous results they had on the super close islands. And so, yeah, for the corals, that's it. And then my supervisor, Angie from Blue Finance, is the coral expert. And for the fish, so I had to train to identify them. So same, I had a bit of experience in that from my post internship in Indonesia. But still, it's a lot of species. So that's why I chose to focus on some specific fish groups to be able to identify them for sure with confidence. And that's why it was the biggest limitation in terms of number of species I could identify, because as you know, it's the highest marine biodiversity on the planet. So it takes years to be able to conduct a full assessment of the whole area. So yeah, and then Denmark checked my results as well and told me it was quite okay. Like, he validated the results because he has more than 10 years of experience in that.
It's quite impressive, yes. And could you go back to the slide 16? I would like, yes, on I think it's the different sites. Could you explain me more? Yes, that one, those different to assess I guess, the diversity of the substrate or the complexity of the substrate. Yeah. Could you tell me more, yes, on an ecological perspective, why separating those 4, 3, 6, 9, 10 different
in 5 different categories. Yes.
5, okay.
5, yeah. Some pictures are for both the same categories, like for 0 on the top left, and in the middle it's the same. It's the same category.
I see 0, 0. Yeah. Okay. Yeah. And so can you explain then? Yes.
So to give you an idea, in the literature, recently there are studies that show correlation between the fish assemblage and the coral reef's structure. So the structure is based on the carbonate skeletons, right, of the hard corals. And what is shown is that with the higher complexity of reefs, so in the category 4 or 5, at the bottom of the image, there are more like holes and areas for the fish to shelter, for example, for nurseries as well. There is more food as well. So yeah, it offers protection and food mainly, and so that's why the fish assemblage are more diverse and abundant in this kind of reefs. Because if you go to the flat like reefs on the category 0, obviously for the lower trophic level fish, they cannot be high. That like the higher trophic level fish can prey easily on them. And same regarding the food sources, it's really restricted. So these are the main reasons why there are strong correlations between both.
Okay, thank you. I think, yes, we can stop here. We'll have other questions.
Yes, thank you, Yasmin. Paula?
Thank you, Cassandra. I think, yes, you did a very nice job. I think you can really be proud of you.
Thank you.
About all the achievement that you did during your internship. Your small internship, but very good conclusions. I have some comments on your presentation. First of all, because you start with the Philippines, it was really a site problems, but at the end, it's an MPA problem. So it will be better to contextualize your work in your objectives, which is the definition of what is the best MPA.
Okay.
So you did it, but I think that you did it on the reverse side. You should start.
With MPA and then
You start to do it with your plan and you say yes, but the end is the MPA, my most important question. So start directly on. You have all the slides, but I think not in a good order.
Okay.
My second question and comment, it's more linked about how you present the results.
Yes.
And I agree that you didn't explain us enough how all your results can really help you to propose the limitation of the MPA.
Mhm.
You have different methodologies, so different tools to identify very important zone and perhaps more strategic zone. And so my question is, do you think about some analysis that could put together all the data that you had in order to conclude about the area that are more important than the other one to be protected? You explain us one result after the other one.
Mhm.
But is there a way to put together all the results and to have a big pictures using all these different data? Even if some you say are less accurate than the other one, but there are still data.
Mhm.
So at one moment I say, okay, the mandatory is not important. So why she present the mandatory? So it's still an information. It's still very important that you did it. So is there a way to put all together all these data?
Together. I mean, for me, it's about all the parameters together, as I said in the discussion, meaning that, if we run statistical analysis, for example, to see the correlation, to try to explain why this area is that way, etc., we could go further of the discussion, obviously. But here, for this MPA, for example, to be completely honest, I was not the one deciding, and it's based mainly on humans. So what they would agree for the MPA to be implemented in that area. So the idea that what I discussed with Nicolas Pascal, so the head of Blue Finance, is that we start small with those areas that fishermen are willing to.
I understand the strategy, but more you're strong on your data, more the people will follow you.
Yeah.
So how you show all the data to the guys,
Yeah.
or you try to see, okay, take in account this data, we have this image of more sensitive zone, more vulnerable zone, more very rich zones.
Yeah. So what I showed during the meeting, is that what you want to? Is that what you mean? Okay. I showed the core results mainly. because I didn't have the fish results at that moment, to be honest, during the first meeting. I was still in the process of processing all the data. And so we showed the areas with the highest coral coverage. And the thing
Hard corals.
hard and soft corals.
Okay.
Both. And the healthiest as well, because we talked quickly about the dead corals. It's highly related to human activities, because what maybe we don't really realize on the maps here is that the main island is at the south, so it's much easier for people to come there. And so that's why there are more damaged corals, for example.
There is village, there is zones with more people than the other on those islands, for example.
On the studied islands,
Yeah.
there's no one.
No one in the 3 islands.
Yeah, it's
Inhabited. Okay.
inhabited. So everyone comes from the mainland of Mindoro. You can see. Yes, here. Okay. So these are the super small 3 islands. Everyone is living here and coming here. So there are more pressures coming on the bigger island here than on the rest. And same, we were working on ghost nets at some point, and we observed much more ghost nets in this area as well. Yes.
So I didn't have the answer how to put together.
I want to put together that. Listen the question kind of, because to me it's all together.
Maybe we can go to Aldine and come back again. Aldine.
Okay, so Cassandra, thank you so much for this wonderful work. It's so rare to go from A to Z during a master project. And honestly, here you bring really something to this place and to these people. And I'm sure they bring you a lot too. So yeah, congratulations for this amazing work. So I have a few curiosity questions. So during the Mantato methodology, in a really biodiversity abundant spot, how many maximum fishes you will be able to identify?
Mantato?
Yeah, with the
No,
Mantato.
it's not about fish. Because
Okay, it's just the coral,
substrate,
sorry, the
yes.
substrate. Okay, but okay, and how many different substrates do you have?
So here we just choose to work on just the hard corals
Yeah.
and soft corals,
Okay.
because as you can imagine, it's hard to remember everything.
Yeah, I agree.
So the smallest, the simplest, the more accurate data we have. So we choose to just focus on estimating the hard coral coverage and soft corals, and that's
Okay.
it. And in most projects with Mantato, they just work on crown of thorn, for example, so they just have to count the number of crown of thorn. So quite easy to remember during over the 2 minutes interval.
Okay, thank you. Did you try to analyze with the coral diversity? So the massive, do you have the data for the difference between the massive and the branching coral? And have you performed the analysis when you are doing coral identification? So did you perform some analysis? Different side, ranking versus.
During the identification,
Yeah.
Yes, we have. So I didn't present it here because of configuration, of course, but yes, I have it in
Okay.
the annex.
But
But yes, we did investigate different morphological differences between the corals. Okay, so mainly branching corals, for example, in the area.
Okay, thanks.
Yeah.
And then what?
Last question.
Well, last question. So what was the most striking data for you to help orienting the development of the NPA? It's coming to Paula's question. So what was the most striking data that you highlight?
Yeah, for me, it's of course the coral coverage, but the fish is really important as well, because what I observed in the water is that, for example, in some areas you have a high fish biomass. There are fish everywhere, but it's the same species. If you know the Niger, maybe the bigger fish, in some stations it was everywhere. But so it means that the ecosystem is highly imbalanced, for example. So I would protect like the most healthiest ecosystems, or where there is a high diversity. So the diversity is a really important parameter to me. Like if we look only at the biomass, it's not relevant enough in my understanding and my opinion.
Okay, thank you.
And just before the break, last question with Isaac.
Thank you. I want to say really amazing work. But I had a question for you regarding the MPA creation. Do you feel, in your opinion, that it was rushed? Do you feel that you didn't have enough time to actually analyze the big picture here? And I guess, for example, when looking at the fish species, like you said, you notice you're obviously identifying the most common species, but what about the rare species that you're not noticing and potentially missing, I suppose, is what I guess what I'm asking, do you feel it was necessary to have more time, or do you think it was justified based on what you did?
For me, of course, it was rushed. If it was based only on my results, it wouldn't be relevant, I think, because we would need more studies, of course. What I want to mention is that in the 3 stations I showed that with Blue Alliance, they conducted the fish surveys already, and they did notice some interesting species. And this is in one of the stations that we chose, like the station was located here in that area. So that's also what supported this decision of having this delimitation of the MPA, and it's not implemented yet. Anyway, so I might change in the future, maybe. So yeah.
Thanks. Thank you. Congratulations again. And so we take a really short break, less than 10 minutes, because we're coming back at 45 with Isis and Victoire. Okay, 9 minutes, 8 minutes break.
Oh my God.
We're all here. Great. So, thank you to be back. Hello, Isis. So we'll start with Isis now and Victoire to finish this afternoon. Isis, you have the floor.
Thank you. Hello, everyone. My name is Isis Almeida, and today I want to show a different facet from scientific research. And I hope by the end of this presentation, you'll see the importance of it. So my theme is strengthening alliances for scientific research, the role of social media and sensibilization actions. My supervisors were Professor Paulo Curla, that is here today, no pressure, and Dr. Eric Ratzinger. So my presentation outline will follow the regular one with introduction, materials and methods, results. In conclusion, the only thing that's going to differ is the discussion, which will be substituted by analysis, limitations, and perspectives. So I would like to start out the introduction with the social media context with a phrase from Krishnagiri, which is a specialist in social media, that says, "The democratization of information that social media have brought to our society has significantly shifted the way individuals communicate, interact with each other, and also how knowledge is shared and exchanged." Indeed, most studies shows that some of the benefits that social media has brought are connectivity and bonding, community building, so a sense of belonging to a group, efficiency in spreading a message, engagement in social and political movements, between others. One might not at first make a connection between social media and science, but Abadia says that these platforms can help scientists to build networking abilities, share their work to a broader audience, receive instant feedback. Another point is that social media also influence behavior of a society, since it improves the public's understanding of environmental matters and also encourage them in revising their own actions and behaviors. When talking about sensibilization actions, some of the benefits that we can name are how easy they can connect to how easy they can capture public's attention, and how effective they are in boosting their interest on specific matters. They can also promote interdisciplinarity and enable collaborative efforts between different actors that have a same common objective. We can also see that usually this type of events, of outreach events, deal with interactivity, which is a very dynamic way of learning. And there's studies that shows that there's a strong link between the active engagement, this type of active engagement, and the level of awareness. Talking about awareness, Castillo and Rojas, which are experts in education, explain that sensibilization actions are pivotal to blossom the environmental awareness in the public. That can be described as not a way of learning the concepts concerning the impacts of human activities on ecosystem, but to understand and use this knowledge to see where we lie and determine how we arrive to these circumstances and propose alternatives to change them. So the organizations that are the object of this study is LIAHOPSE, the Associated Laboratory on the Theme Responsive Organisms and Population to Environmental Stress. They are a collaboration between the Université Côte d'Azur and the Centre Scientifique of Monaco. They have three main study axes, which is marine biology, medical biology, human and social sciences. And some of the partners are ECOSIS, ICANN, ICN, between others. When talking about the Federative Research Institute of Marine Resources, or IFR-MAHES, it's also under the scope of the UCA, but they are exceptionally focused on the studies of the marine environment. Some of its partners are CEPAM, GRADEC, LADI, and MSC-MAHES. So their main objective is to use innovative research and scientific excellence to solve modern-day issues. Being stated that, I would like to propose the following questions. The following question: how can we increase the interactions between marine research centers and other actors in the field while also increasing the internal and external visibilities? For that, I would invite you to take a look back on what has been done in the past years by the last interns. In 2021, Julie Gastaldi proposed a questionnaire for the LIAHOPSE partners, in which they stated that they were in great favor of this entity being present in social media. In 2022, Lara proposed an assessment of the efficacy of the visibility tools, and in its conclusion, she stated that the next intern, which was me, would be interesting to assess how people perceive social outlets for research. And on my part, I decided to add the sensibilization events to this hypothesis because of literature review, and I saw how important it was. So here I share with you my hypothesis. Evaluate if social media and sensibilization actions can first strengthen ongoing alliances and promote new ones in the marine research field, and serve as an instrument of knowledge and awareness to the public. For that, my materials and methods consist on first social media. For LIAHOPSE, we picked Instagram as the social platform for data analysis. With 2.3 billion users worldwide, it provides 58% more engagement than other platforms, such as Facebook, for example. Instagram Insights provided the metrics such as total followers, average accounts reached by posts, and average accounts reached by stories. For the website, we use Google Analytics, which provided a user's overview, acquisition channel, and language preference. For MAHES, the social media platform picked was LinkedIn, with 310 million users worldwide, and with 40% of which engage organically with at least one post per week. The LinkedIn Analytics provided total followers, new followers, page views, unique visitors, impressions, and engagement rate. For the sensibilization actions, we performed 2 surveys, both in which were performed during Ocean 21 events. The LIAHOPSE survey was done for scholars from 2 primary classes and 1 middle school class during the intervention in the Bibliothèque Raoul Mille. And the intervention was done with the proposition of 3 workshops on the themes fluorescence, regeneration, and symbiosis. 5 simple questions were done before and after the workshop with the objective to assess the children's comprehension on the themes explained that day. Since we already saw previously the importance of interactivity, we used puzzles and charades and microscopes to engage the children's attention. For IFR-MAHES, the wide public survey was done during World Ocean Day And it was done with 30 interviewees or 30 visitors of the IFR Mahis booth in the village, awareness village in the Promenade des Anglais. The survey was provided both in French and English to capture a more broader audience. The objective was to assess the public's opinion on the research centers being present in social media and how they perceive sensitization actions, such as the one that we were dealing with that day. To start out my results, I want to show you some numbers. So for the Lia Hops's Instagram, we saw that a little bit less than one third of its followers were reached by stories, while more than 2/3 of its followers were reached by posts, so more engagement on posts. And during the internship period, there was an increase of 42% of followers. Now, for the acquisition channels of the Lia Hopse website, so where did the visitors came from? How did they access the website? You can see that social platforms are in third position as main source of visitors, with 6.8% coming after organic search, which is when you type in Google and it appears as a listing, and direct, which are unknown sources, so you can't really track where these visitors came from. For IFR Mahis LinkedIn, here we have a graph that shows the page views during the internship period. We can see 2 peaks marked in the circle, in the black circle, which accounts for May and June, which were the most active months of this during the internship, with publications, events, conferences, and thus more content posted on social media, which provided more engagement. So here we can see that there is a strong link between the activity on the platform, the page views, and the engagement rate that follow the same pattern. For the wide public survey, 2/3 of the interviewees, when asked if they already knew that 8th of June was World Ocean Day, they said that they didn't know about this important date. So 66% say they didn't know it about before the date, before the event. On getting asked about how did they get to know about this event, social media is in the second position as to how people got to know, showing again the importance of social media in sharing information, only surpassed by passersby, so people who were walking on the street and stopped to see the event. Now, some other numbers that I would like to share. 100% of the interviewees show that they thought that sensibilization and scientific events are a great way to scientists connect with the public in a more personal way. 93% of them said they would participate again in similar events. When asked about how they feel on research centers being present in social media, 100% said that they believe it's important, but only 30% of them actually follow research centers in social media, which is strange. So, for the scholar survey, we can see that a graph shows the percentage of correct answers before the workshop in light blue and after the workshop in darker blue, which we can see there is a clear increase of the correct answers chosen after the workshops were performed, showing a clear understanding that by the kids of the marine teams explained on that day. So what does these results show us? For social media and website, we can see that the IFR Mahis and the Lia Hopse had a growth in their community, not only when talking about followers, but also on the engagement and how people perceive the scientific information shared. More engagement share more results in more visibility, and more visibility can increase a brand's awareness, establish credibility and expertise, and also promote more collaborations and connections. For the sensibilization actions, we can see the importance of the organizations, Lia Hopse and IFR Mahis, the Ocean 21 events framework, by sharing knowledge with the public in a more immersive and dynamic way. We can also see that there is a positive view by the public regarding outreach events, since 93% of them would return and participate again on future ones. And the importance when dealing with kids of an adaptive speech to the age gap we're dealing with, and the use of the correct medium, so interactive mediums, are a deal breaker for the success rates of their understanding. For our limitations, sadly, the activity is not regular, which means that when there's no science communications intern, the social media is not active. The IFR Maha's website was not contabilized in this presentation because sadly we couldn't track the Google Analytics of it, although many efforts were done on that matter. More studies are required, for example, to link why people think that it's important for institutions to be in social media, but they don't follow them. So why is that? For our perspectives, we would be interesting to find a way to motivate partners of both institutions in posting on social media and websites more often to continue this engagement and not be a lack of activity as it is right now. Also, the implementation, obviously, of the tracking for the IFR Maha's website would provide valuable information for engagement, visitors, and acquisition channels, so we could also see how social media and sensibilization events affect that. Also, it's important to note that the presence of researchers and scientific centers on social media can debunk false information, which is a very common problem with social media these days. Lastly, I would like to conclude my presentation on restating the importance of Lia Hobson and IFR Maha's on the local research development on the marine field, and also in the sharing knowledge with kids and the wide public. I would just like to go back to my first hypothesis, but to strengthen ongoing alliances and promote new ones in the marine research field, which can be partially confirmed, since the data on social media and the Ocean 21 events show that there was an increase of visibility and promoted partnerships, for example, with the Vietnamese. For new alliances, only the future will say. That's why we say partially. For our second hypothesis, to serve as an instrument of knowledge and awareness to the public, we can see that it is confirmed since both surveys shows an increase in knowledge and awareness. Lastly, for the next intern, as Lara did last year, I think an analysis of other social media platforms and sensibilization events would provide more comprehensive results. I would like to just end with a take-home message to make you guys think. How can we expect society to change its behaviors and actions towards the environment if the scientific knowledge is not shared or seen outside the research field? And that is it, guys. This is my reference, and I thank you for listening.
Okay, let's do this differently. Let's start with you, Aldine, and then we will continue with you, Robin.
Hi, thanks for this work. So my question is to which public your survey was addressed?
The wide public.
You talk about survey, so can you just redefine?
So there was 2 surveys done.
Mm-hmm.
The first one was with the scholars. So it was, oh, sorry, I put the wrong one. The first one dealt with 3 classes of, oops, is that it? No. Yeah. 2 primary classes and 1 middle school class. And so kids, and they came, we performed the workshops. We performed the workshops, we did the 5 questions. We assessed how they responded. We did the workshops, and then we assessed them again. For the other survey was with wide public, so anyone that would stop at our booth and was willing to just answer the question. We just had some papers with like a QR code that the person would scan, and they would just do it on their phone. We also had like iPads available if the person didn't have any phone with them.
Okay, so don't you think that your surprising, so what you call your surprising conclusion about the fact that apparently people think that it's important for research institute to be on the social media, but they will not follow them, just because you exchange with a public that was maybe not specialized enough to go to such websites? Or do you know why they say that?
Yeah, some of my conclusions on that. First of all, we had a variety of age gaps on this wide public survey. So people ranging from like people our age, like 25, up to like 75. So the first thing that I would come to mind is that older people tend to not be more present in social media. So that's why maybe they don't follow anyone. The other thing is that obviously this is a small survey. It was only 30 people, so I cannot really assess that it is true. Like, oh, a lot of people think that being present is important, but they don't follow. And also, maybe that's why I said that it would be needed to do a more research in depth about like why people think that and why they act like that. Maybe there's not much knowledge that these institutions are out there, you know. And yeah, that's what I thought about it.
So if you have to do something
Yeah.
to push the efficiency for collaboration and for to optimize these communication things for science and so on, so what you will do now?
I feel like part of the conclusion of my work is that, for example, events like such the one that we did for Ocean 21 can capture a lot of public that may not consume scientific information on a daily basis. So since we saw that most people were just passing by on their walk, on their way to work, or something like that, and they just stopped and started seeing all this work, these microscopes, these fossils and everything, these NGOs just sharing. I feel like that's a great way to like, hey, do you want to know more? Like we have a website where you can check our projects. Hey, we have a Instagram platform where we can share like news and, you know, that type of thing.
So means using that human connection.
I feel like yeah. For external visibility, I think it's really important to connect personally.
Okay.
And obviously, like on an online level, the more you post, the more people see, the more people engage, the most people retweet, and you know, it just grows.
Okay.
I feel like.
Thank you.
Thank you.
So let's go to Robin and Yasmin.
Thank you, Isis, for the presentation. This is very, very far from my expertise, but I mean, it's very interesting, and that's really, I think, an important question on how to disseminate the scientific knowledge to the public. It's, I think, it's under, I don't know, it's overlooked by scientists a bit because we are already doing a lot and we don't spend enough time on it. So I think it's really important to have at least some study or some people that try to do the link between the scientists and the public. This said, the count that you show, were there actual counts or proportions, or I think there were, because there were no units, and it's like sometimes I don't know, do we have the slide number? Yeah, for example, on the slide 8, it seems to be accounts, so I guess it's
Is it?
What?
Slide 8, you said?
yes. Okay, the one, well, on my laptop it's 8. I downloaded the PDF, so it's probably the 9.
Oh, okay. Wait,
Or
it's
10, not
because
even after that.
yeah,
It has changed a bit apparently.
no,
Okay,
it's
even
because
after.
it's with animations and without animations.
Oh
So
yeah, there's no animation. Yeah, this one.
this one?
Okay, so these are actual accounts.
Yes. So this means that so Lia Hobson now have 89 followers, but at the end of my internship, it counted with like 79, 80. So that means that every time someone posts a story, around 26.8 of these followers or these accounts that follow the Lia Hobson Instagram receive or see. Whereas when posts are made, 74, so almost like all of the followers are reached and can engage with it. That's what I meant.
Okay, that's what I understood. But and so if you continue, so I cannot give you the slide number.
Yes,
If you go
sorry.
to the Mares, to the IFR Mares LinkedIn.
Yeah.
So here it's page view per month, or on the day that is displayed at the bottom.
So I put the parameters from March 6th to July 21st, which was the internship duration. And here it's kind of like per month. It just here it says March 6th because I started on March 6th. Otherwise, it would just be March 1. And it just goes by month. So this is a by month review.
Okay. So I mean, here I think this is enough to make my point. Here in April, you have 2 people basically that visited the website.
Yeah.
Do you think that's among the months? So that's 1 every 15 days. I think it's quite few, and even if it's 1 per day, that's very low. So my question, I mean, if I interpret it correctly, but my question behind it is, how do you think? Because there seems to it's difficult to disseminate the scientific knowledge and to raise the interest with, I mean, robust content, interest content, regular content, publishing, etc. But given the low numbers that you have here, don't you think you had a higher impact with the in-person or the live, I don't know, you call it the in-person event, like the Ocean thing, when you actually met the people, than from what you post on the social media?
Yeah, obviously the numbers are not big. The follower count for the IFR Mahes, I think, are like 200 and something followers, which means that if we don't have much posts per month, really people don't visit the website. Not the LinkedIn page. Obviously, I feel like in-person contacts and the events and the outreach events that we promoted were more successful in the sense that reaching more people, but I don't think that I don't undermine the power of social media. I feel like every time, the more you grow, the more you get to see, and that's my point of view. At least I feel like it's obviously less impactful at first sight, but I hope that with my context, I feel like we could see that it is an important tool. It can be really important.
I'm really not discrediting your work and the social media.
It's just to defend it.
Yeah. No, I know it's very difficult on the count and get this activity, this regular activity is it's a full-time job really. What did I want to say? But the issue with social media, I mean, with social media in general, is that how do you get to know that you reach new people that are not already aware of what basically you are doing? Because I'm more likely to follow the lab page of a friend or a colleague or someone I work with than I don't know. I mean, I'm likely to do that, but I'm already aware of what they do or anything. So is that dissemination, or is there a way to actually count the people that are unaware, following the new people, etc.? Are there tools for that?
Yeah, definitely. There is a okay, I have in my annex actually the count for unique visitors, which means not the people that consistently go on your page multiple times, but the ones that just go. So it counts like each unique visit, so not recurring visitors. That's what I meant. So obviously the numbers are low, but we can see that there was a number again with the same May, June, meaning that they had during the most publication months, we had more visitors that never visited the website before. I think that's what you meant, like people.
Yeah. No, that's what I meant. Yes. I mean, well, you can still not discriminate between the people that basically like people from the scientific field to people that are just like new.
Yeah.
I mean, not
There
scientifically aware or interested people. I mean, that is probably, I don't know. I don't even know if it's feasible.
Yeah,
Just
there
a question.
in Google Analytics, and I think in the LinkedIn Analytics, you can, there's a section where there is demographics of your public, and you can see like where they come from, their language preference. For example, that's something that I also said I just didn't include. So we could see that there's like a lot of English speakers as well as French. That's why it was important to translate the website. And you can see also like their field of interest, so like educational or really scientific or medical, but it's really difficult to track because, as you could see for the acquisition channels, the direct one, which is unknown sources, is like people who have blocked their IP. So you cannot really know. You can know from some of them, but not a complex like overview. You know what I mean?
Yeah.
So, yeah.
Okay. Thank
Thank
you.
you, Robin.
Thank you.
Yasmin, you're mute. Your microphone is off, sorry.
Yes, I was saying thank you for the presentation. I think you did a good job in presenting something that is unusual indeed in our field, but essential because I think each of us have somehow social media page, or we access social media at one point or another. So it was, yes, thank you for that. You did somehow also a complex job because there was two aspects. There was the aspect of developing a questionnaire and the aspect of analyzing the result. I mean, all those the two different social media from Mares and from the Elia Rops. So I have one question regarding the development of the questionnaire. What would be the most bias or maybe critic point you would do from what you have, from this questionnaire? Because I don't think I had seen that. And what's from your point of view? Because it's always, I'm not in social science, obviously, but I know that there is a way of asking questions to not. So, can you tell me more on what type of research you have done and how you have decided, without going too much in detail on those questions?
Definitely. For the scholar survey, we decided to do short and simple questions first because it's easier for the kids to understand. Obviously, they have to understand the question well to answer. And also, they were based on some of the questions that were done last year by Lara in the same event. It was literally the same event with the same workshops done with, I believe, the same age gap with primary school and middle school. I think I just added like one question that was the invisible lights because I thought that was like interesting to just make them question like, wow, what is invisible light? And yeah, the focus was just maintain a simple question, so 5 that would also not take a long time to perform since we had a little very short time and the focus was on the workshops being done. And for the web public survey, I didn't include that because I thought it was like a lot of information. But I put it on the annex. Here it was done on a Google Forms type of spread. When I was developing it, I also, of course, as supervisors, ideas and tips. And we all thought that really we couldn't extend too much the questions because once someone starts to answer and see that it's like endless questions, people just lose interest and stop answering like, oh, I'm not going to finish this. So we tried to condense as much as we could, but really we wanted to deal with a lot of stuff. So I tried to kind of divide between assessing how they perceive the social media, what kind of social media they used, if they use social media, again, if they follow the research, if they knew about the researches that were being presented, the research institutes that were being presented that day, and then to assess how they see the outreach events. So if they like, there were questions like, oh, what did you learn today? Can you summarize in like 3 questions or 3 words? And just to kind of get an overall picture. It was not like very in-depth questions, since it was just like meant to be more answered, you know? It's not,
Okay, yeah. Okay.
I hope this
Maybe that, yeah, maybe that's something I had missed. So for both questionnaire, it was around a workshop for both of them.
yeah, so
It's the same, was the same workshop. It was Ocean 20.
yeah, the Ocean 21, it was like a 12-day event since 2021, promoted by the Denise, which is like a week-long. It was supposed to be a week-long, but usually it takes more than a week, with like interactions, interventions, like exhibitions, movies, all about ocean literacy, ocean conservation, with NGOs and everything. So one of them were done by Yahopse, which was in the bibliothèque with the scholars, with the workshops done by the Yahopse partners, so PhD students that came and performed the workshops. For the IFR Majes, it was awareness village one day after World Ocean Day, because the day was raining, and so they moved. And it was like a bunch of booths on the promenades, and the person would just go and like visit, for example, the Denise or an NGO, and then there was IFR Majes, there was MSC Majes as well, and they would just get to know there was not like any workshops being done, but there were people presenting their studies and their works. They had like microscopes so people could
Okay,
see stuff,
yeah.
and like fossils that we
Yeah,
could
yeah, and you interviewed the people coming through. Okay,
after
so
they saw. I was like, oh, can you have 5
So
minutes?
okay. Okay, so concretely, I mean, I know you have proposed a what's next, but I'm still, yes, not, can you go back to this slide?
This one?
Yes, the what, yeah.
Or
and then
the perspectives?
No, the what's next, you had it, yeah, no, it was good.
Yeah, okay. Mm-hmm.
Yes, can you tell me how, what concretely this would lead? Because I'm still confused on the objective really of your questionnaire. And because I had the idea of trying to see what, I mean, you made the introduction on how important social media is. And then you had a questionnaire for the general public that was kind of linked to social media. For the younger one, it was obviously not. It was more the question about animals. So I'm still trying to understand what exactly could be done to really promote, because one important key aspect was to transfer the knowledge from scientific to general public. So concretely, how this would help to better convey this message by doing this next step.
Yeah, so the next step for me, I propose, since the Instagram and the LinkedIn pics were done because these platforms were already been used from the other interns. And for example, IFR Majes didn't have any social media. They only had the LinkedIn page, so it was the only option. But maybe LinkedIn is not the best option. So it would be nice to maybe try, the next intern try to assess either implementing a new type of social media, or assessing if there is an interest on other type of social media, or if people already feel like LinkedIn is enough. For Yahopse, we have the Facebook and the YouTube page. I think IFR Majes also have a YouTube page, but it's not really active. And that's what I meant for the next intern. But I feel like the conclusion of my research would be to know where to focus your efforts in. As Robin said, like, the scientists have a lot to do already, and I don't feel like they think that social media is their priority. And I wanted to say that it can be a priority, you know, it can be something also to look for, you know, something also important. So know what to focus on, like which media to use. I think it's like essential, you know, to just be productive on the sense, be effective on
Okay, so
spreading the message.
you're still convinced based on your result.
Yeah.
Thank you, Isis.
I work.
Okay, Isis, thank you, Yasmin. We will
Thank
finish
you.
on that note. Congratulations.
Yes,
Isis.
congratulations.
And here we go. Victor, you ready?
May I just say something? I would like to thank also Sarah,
Alors,
because Sarah
Yeah.
was also involved
yeah.
in the internship, so I would like just
I
to have
thank
that.
everyone in my report. Sorry for the thanks for the representation.
Okay. So we'll conclude the day with Victoire. All good? And you have the floor.
Passer celui-ci.
Oui.
Okay. So, hi everyone. I'm Victoire Esneux, and I'm going to take you to a tropical island, which is Zanzibar. So, Zanzibar is an archipelago of 3 main islands, which are Ungunja, Pemba, and Mafia. Mafia is not included in this study. So, the Zanzibar archipelago is along the Tanzanian coast, and Tanzania is an Eastern African country. Over the last 20 years, tourism considerably grown because of this growing interest and attention to coral reefs. But this induced an increase in boat frequentation over this marine ecosystem, which led to disturbance and impact on coral reef populations. So in other points, the artisanal fisheries are prevailing in Zanzibar because most of the boats are unable to reach the exclusive economic zones. So therefore, the fishing activity is concentrated into shallow and coastal waters where the coral reefs are. So in response to a growing population, the coastal waters became overexploited, then overfished. And in addition, despite the existing MPAs, so marine protected areas, there are still illegal fishing activities ongoing, such as the beach sealing, which is the dredging of the bottom. So the Eshimu Bahari project aims to address these issues by protecting coral reef and their high biodiversity, and because of their good and ecosystem services for the local population, such as many livelihoods with the fisheries activities. So this project is developed by WCS, so Wildlife Conservation Society, in partnership with the Zanzibari government and its growing blue economy. This project is funded by USAID, which is a public independent agency under the guidance of the United States, just to mention. So the project is divided in 5 steps. So the first step and the second step are the ecological surveys and fisheries data mapping that will help gather valuable information about to collect valuable data. The third step and fourth step will introduce side-by-side monitoring and fishery management plans with the local communities. Then the overall main goal of this project is to implement no-take zone
within already existing MPAs, as you can see here. Sorry for the quality of the map, but here we have several MPAs near, except in this part of the island. So what is a no-take zone? A no-take zone refers to this designated area within an MPA where all extractive activities are prohibited or strictly regulated, such as collecting or fishing. So this no-take zone aims to establish marine really highly surveillance protected area in order to for the marine ecosystem, such as the coral reef, to restore. So we began this project by the benthic substrate assessment of the Tunka area, which is here. So Tunka corresponds to the Tumbatu Marine Conservation Area. And this site is actually of great interest because of the presence of coral reefs, sandy beaches, seagrasses bed, rocky shores, and mangrove forests. So just to mention again, there is no active monitoring, and these ecological surveys with the benthic substrates assessment will help the mapping of a large range of marine substrates. So this exploratory and preliminary study aims to seek if sea urchins' abundance can be considered as bioindicator of living marine substrate health, but more especially coral reef health in the Tunka area. So before presenting my results, this is a little overview of the method that we use to collect data. So we use the photo quadrat method to perform the benthic substrate assessment. So we have this GoPro camera attached to this metallic frame that in each station we would put down 3 times to obtain 3 replicates, and in order to increase the accuracy of the data. So here, as you can see on the top left map, that's the station where we're going from the shore until 10 kilometers offshore, sorry. And each station was 1 kilometer apart from each other, every station. So now for the pictures analysis, we used the free website CoralNet with the 25 stratified random method, which in other words is here, as you can see, you have 25 either square or circle randomly spread across the picture. And the goal is to identify per each inside the square which marine substrate are you observing. So, yeah. So after obtaining our data, we were able to creating different maps, and this is the one that I selected for you to see the extent of the sand coverage over the Tunka seabed. And so here is the Tumbatu Island, so this island, and the red rectangle corresponds to an area where we have no data. So I forgot to mention it in the methodology, but when the depth was more than 50 meters, we weren't able to retrieve, to collect any data. So this is an old area where the depth is above 50 meters. So referring to the legend that we can see here, the more in this map, the more darker the station is, the lower the sand coverage in percentage will be. So that indicates the presence of other marine substrates. And as you can see here on the other maps, you can see that we can get matches because here the coral coverage and sea urchin coverage matches the places where we can see a lower percentage of sand coverage. And you might wonder what is this purple layer. This purple layer corresponds to the a satellite image of the coral reef of Zanzibar. So this pie chart displays a summary of the overall tendency of the Tunka seabed coverage. So you can see that the non-living marine substrate cover is dominant over with 69% over the living marine substrate cover in Tunka area with only 31%. But this pie chart doesn't tell us much about the relation that we want to explore between the sea urchins and the coral reefs. So therefore, we performed a regression analysis, and here you can see that the relation is positive, which means that a higher frequency, the more you observe sea urchin, the higher the coral coverage will be. So, however, we must be cautious with this model because of the low model explanatory power, which is only 0.0348, and also the fact that the variables do not follow a normal distribution. So, with that being said, thanks to the p-value, we can this regression analysis can still give us some insight about the relationship between the frequency of sea urchin observed and the coral cover. So, assuming the relation is positive, we gathered every living marine substrate to see if we would obtain the same tendency, the same relation. But here, as you can see, because of the p-value being much higher than 0.05, this model is statistically not relevant because the linear relationship is not strong enough. So we wanted to compare our first results with our positive relationship between sea urchins observations and coral cover with literature. So in this first study, you can see that in the sea graph, that's the relation between sea urchin cover and coral cover is positive. However, this relation is explained by the negative relationship between sea urchin and macroalgae because the sea urchin is grazing on macroalgae, which implies that the sea urchins will will enhance the coral recruitment because it's helped reducing the main competitor of the coral reefs. And here you can see that on this second study that shows us that the sea urchin's density is positively related to juvenile coral density. So just to confirm this study. And as a third study, which is of great interest, we can see that the relation is negative between the sea urchin's density and coral cover. So what is really interesting in this study is that in low sea urchins abundances, the coral resilience is enhanced, and because the sea urchin will be grazing on macroalgae. However, in high sea urchins abundances, there will be a reduction in living coral cover. So it leads us to the conclusion and limitation of the study. So the sea urchin's abundance can be considered as a bioindicator of coral reef health because of the studies that we just saw. But however, in our study, we can assume that the sea urchin is a bioindicator of the coral reef health assessment. However, we cannot validate our hypothesis because due to the lack of fauna data and the lack of references and previous studies in the area. And also we can mention that a few limitations about the data collection and a low knowledge in marine substrate identification when it came to the analysis of the pictures. So for future perspective, the fisheries pattern mapping will give us valuable information, such as the fishing pressure with the environmental factor to take in account in this kind of study. And what also is of interest is to investigate the CoralNet point identification methodology to assess benthic substrates in our dataset. And yes, I would like to acknowledge many people for this internship, and thank you all for listening to me.
Voilà, we're back with you.
Start? Okay.
Yeah.
So many thanks. It was a nice, interesting work. Again, I think that you can implement some data in once in order to have a really definition of the different sites that you are looking for, because you have different data, the sand, the corals, the presence of urchin, so you can try to put all together. It will be interesting to have this analysis to identify the different sites on your area. But I have a major questions about the sea urchin. You speak about sea urchin, but are you looking about one specific species, or you look about different species of sea urchin? And the other question is, why the sea urchin and not another benthic species?
So, because we performed like the benthic substrates, and the sea urchins were assessed. So every kind of species were counted. We were not so regarding the corals, the algae, seagrasses, or sea urchin. We did not assess the species diversity. So we took interest in.
You did not assess, but you can say that there will be multiple species, or there will be just one species on what you saw?
So from what I saw of all the picture analysis, that was mainly one species, but for example, in the seagrasses bed, there was another species. And so we took interest in sea urchins because they, in usually fishing pressured area, they represent the grazer organism that is not fished. And in this case, in Zanzibar, the sea urchin is not fished. That's why we took interest. But we really have this missing data on the fauna.
And my second question is about your result that you present in what I saw in the DIA 9. So it will be 2 after DIA. Yes, this one. I have the feeling that perhaps your linear regression could not be the best regression. I have this feeling because I saw that within a certain number of sea urchin frequency, you have a huge coral cover with something that it's plus minus linear. But then I have the feeling that there is like a threshold where if there is a lot of sea urchin, then you have a very low corals cover. Yes. And this is something that you can see even here in the Mediterranean Sea. So the sea urchin is really say, okay, if you have quite a number of sea urchin, could say it maintains the biodiversity in one side, that it's a signal of healthy reefs. But if there is too much sea urchin, that it means that there is a disequilibrium in the field, in the traffic chain, and then you have this huge amount of CO2. Do you think that you can have something like that through your data? Because you only try to find a linear regression. And I'm just asking if it's correct to only try to find a linear regression.
It's in a future perspective to try other regression analysis, of course.
Because it was my two hypotheses. It's not a linear regression, or there are two different species. Species that are linear with the corals cover, and other that are not. This is why I asked you if there are one species or another one.
Yes, now it's something to consider in the data set, to maybe review all the pictures and decide to identify different species.
Is possible through your pictures?
So with so here you have 300 stations, and so in total the project counts like 4,000 stations overall, and you have to multiply this number by 3 with the picture, with the replicates. So for only Tunca, we had around 300 stations, and the overall number of picture to assess was 900, which means that we could review indeed this picture, but it would take a long time. Of course, it's.
Yeah, I have a last question.
Yes.
Very last question. You say that you try to correlate that with healthy coral reef. My question is, what do you mean about healthy coral reefs? So it's linked to the reef, to the increase of biodiversity, or it's linked to what is the shape of the reef, as we heard before? What is a healthy reef?
Healthy or
healthy?
Oh, sorry. Okay. So a healthy reef is the observation and assessment of different dynamics between all of these elements. We can assess if coral reef is in good health or is in bad health. And sorry.
Okay, so it means if you have all the different participants, I would say.
We need more studies and understanding into the relationship between all of these organisms.
Thank you.
Let's continue with you, Azine, and we will jump with Maria Smith.
Thank you for this talk. So my question is going back on the searching. So you try to answer to why and so on, but are they not going out by night often?
The searching.
Many species of searching, they are popping up by night. So how this can bias your entire analysis?
That's a really good question, and I didn't research on the sea urchins' behavior.
Yeah.
So I don't yet have this information.
Okay. And which other species can you envisage looking at?
What? Sorry?
Which other species, animal species, can you use as a bioindicator?
Grazing fishes. So there is this existing studies. So actually, in the Ngunja Island, but it concerns. This is a study that was done in a strictly regulated MPA and out about the corallivore fishes.
Okay. So when you mentioned that the choice of this sea urchin, so it's also because there in this part of the world they are not fishing it. Are you talking about the fishermen?
Yes.
When you say they are not fishing it?
Yeah, there is no. No, they are not fishing the sea urchin.
Okay, because do you know the other people from the village other than fishermen. Nobody is fishing them, and nobody is eating those animals there.
So actually,
Sure about
not from what I know, but from what I know, there is during the fishing. So the fisherman goes to sea, and we have the fisherwoman that search on the beach the many species. And from what I know, they do not take interest in the sea urchins.
Okay, thank you.
Most of the sea, there is some development of seaweed farming in Zanzibar.
Okay, thank you.
Okay, so we can jump to you, Yasmin. We'll finish with Romain. And again, sorry, the microphone.
Yes, sorry. Yeah, I'll be quick because we all were wondering why, I mean, not why sea urchin, but I had also questions that were already answered about the sea urchin. But can you tell me what it was the main objective of your study? And why do you think it is important to conduct such study?
So the main objective in my study was obviously exploratory and preliminary due to the lack of certain data. And because of the literature explaining this important role of the sea urchin, I wanted to explore how my data was regarding this information.
But I'm still confused what you are expecting, what would be the outcome of such study? What do you hope, what type of information? It's just exploratory. What would you gain from this study? Why doing such study?
So adding the fauna data, the outcomes that we would like to have would be to have the same results than this than the third study that I presented, which explained that when there is low sea urchins abundances, it can enhance the coral recruitment. But if there is too much high sea urchins abundance, it could erode large part of both living. It could have a negative impact on the living coral coverage, which is explained in this study. Is that a higher abundance can induce the reduction of faunal diversity and
I understand this, but why is it important to do that?
In order to assess where in our areas where are the best areas where we have to implement no-take zones.
Where you have to implement what? Sorry, I didn't understand.
Sorry. So the main objective of this really large-scale project is at the end to implement on a, I would say, strategic area the no-take zone in order to promote coral restoration and for the marine ecosystem to heal, I would say, and to retrieve, to recover its biodiversity.
So it's to identify zone where they should implement a no-take zone, and to do that, you assess the effect of sea urchin on coral. There's one thing missing.
Yes. Sorry. Okay.
Just try to phrase, because I think it is important just in your thought process to try to not really to try to find something that it was found in another study, but it's just to help you to really to go through those different thought process that are essential to know why finally you are doing this small part of course, a wider study.
Yes.
And I'm still, yes, missing the phrasing for that. That's it.
Yes, sorry. Yeah, but
No,
it's
no.
as you explained it.
Okay. Yeah, I think we can stop here. I'm trying to see. Yes, can you tell me? I'm not familiar at all with CoralNet, so I understand you take photos, then you insert those photos in a platform, and it's then, yes, explain me.
Yeah, so CoralNet is a free website where you
Yeah.
can create your project and you upload as many images as you want. And you create your label sets, which are, so the label set has been created by other projects, and you can either create your own label set or retrieve already existing label sets from the platform. And therefore, you create your label sets, and then you move on to the analysis phase, where you choose which methodology you want to use for identify your pictures. And here it's which we have chosen the 25 random stratified points. So, for example, you could ask to increase the number of points or decrease it. You can, for example, just have 9 points on the picture to identify. 9 points, 9 circle or 9 square, or you can have 49 square or circle.
Okay, and that's not what you have used to identify the sea urchin, right? Or is it?
So I just counted the sea urchin per picture.
So you did visually from your picture, but not for the coral. The coral is with the website, and the sea urchin was you counting them on the photos, correct?
Yeah.
Okay. Well, I will maybe ask another time because we are. So I don't know what a label set, but it's okay.
Sorry.
I'll have another. I will have. No, it's okay. I think I had the answer, and it's fine. Thank you.
Yeah. Sorry. I have it in the annex. Yeah. So here you can see that you have. It's just like assessing, like identifying if in the circle you have rubble, if you have dead coral, if you have shell hash, sand, sponge, not sea urchin, sorry. But that's a really good point, is that in the label sets, the echinoderm label was implemented.
Thank you.
So that's one of the limitations to, in a perspective, to change, in the future to change the label sets.
Okay, thank you, Yasmin. So we can go to conclude with Robin. So last questions, Victor.
Thank you, Victor, for the presentation. I'm going to be brief. First thing, I was happy to see some ggplots. I don't know if you discovered with me, but I was happy to see them.
Yes.
So you said there were 300 sites that you sampled with 3 pictures per site. Is that it?
Yes.
Yeah.
Yes.
Yeah. So and you did all the image analysis.
Yes.
I mean, that's
yes.
creative work to do. I really agree. I mean, there was just actually, no, while we were on that, did you have any surprise, or do you think this data adds anything compared to the satellite mapping of the coral reef? Because it seems to match pretty well. And should you use the coral mapping, I mean, the satellite mapping to select the area, or did you have any surprise between the two? I don't know.
Actually, there wasn't many surprise. I'm sorry I didn't put it in the annex, but if you take the map of coral coverage and you seek for the space between the island and the purple layer, you will have mainly highly presence of seagrasses, and which we can find in the literature is that the seagrasses bed have an important role for the against bioerosion, and so they help retain sediments, like avoiding, like to keep them from going to the coral reefs, and therefore inducing disturbance. But so no, I didn't have any surprises. Maybe the surprises came from the picture analysis, where during in the picture you would see a sponge fit, and then as you can see on the last image, like for the benthic substrate assessment, you would just have the picture of the sand and the sponge fit, but the sponge would actually be of a really great size, and this is the only surprise that I encountered in my study. But other than that, it was pretty well matching this purple layer.
Okay, thank you. I really agree with Paola. I think it was Paola regarding the relationship. When I see this data, I really see no linear relationship at all, and whether like I don't know. Log normal decrease or something like that with a peak or something. And I'm also wondering what happens. I mean, I see that in the data you included all the sites with zero coral coverage. Does it make sense to include in your analysis the areas, the sites where basically you had sand, where you less likely have sea urchins, or I don't know? But I saw you included them, but does it make sense to keep all the sites where you have absolutely no coral to look at the relationship between coral and urchin?
No. It was just for you to have an overview of the extent of the non-living matter, the extent of the sand coverage. But obviously, the more interesting coverage are regarding the living one, the living marine substrates.
And one very last question, just again regarding the frequency of sea urchin. It's the number of urchins that you had on the picture, because I saw that it goes from 0 to 15, something like that. Where is it?
Yes. So. That was
a question.
Yeah, that was a question.
Yeah.
Okay.
Yeah.
Can you ask again the question, Robert?
What was the unit of the frequency of sea urchin observed on slide 910? So it's after that?
Oh, it's
Yeah.
It was per unit.
Yes.
Per unit.
Yeah. Like
Per picture?
Yes, per picture. Sorry, I didn't understand the question.
So because it seems if you look at the studies from the other location that you have, I mean, one does not have unit, but at least in Taiwan and Kenya, it's sea urchin density per square meter. I don't know if your picture was 1 square meter, it was probably around it or maybe even less. So the densities that you have seems to be quite high to much higher compared to what they had. So I'm just wondering whether you had compared the actual densities of sea urchins that you had in the area compared to the others.
No, that was not included in the studies, and it's one of the main environmental, and it's one of the factors that needs to be assessed in future studies, in future perspective.
We're going to have to finish here because it's already more than 4 p.m. So thank you for congratulations again, Victoire, pardon. Also, I wanted to say, but also to Erika, Cassandre, Isis, and Victoire for this afternoon. And thank you for the jury members for your questions also, and being here and spending the whole day with us, Robin. Merci beaucoup. Robin, Yasmin, you stay with us, so we take some minutes to debrief. You have to run for maybe I know what. Robin, okay. And I don't like for this morning.